The next topic is DNA, which has really revolutionized forensic science. I want to do a little review of DNA and its structure first, just to kind of refresh your memories what that is. So we have parts of our DNA structure that are the same. They're similar to each other. It gives us, for the most part in our world, 2 arms, 2 legs, 10 fingers, 10 toes, 2 ears, all that kind of thing. And there are about 10,000 of these units called genes that are what code for certain proteins and the genetic traits of our cells. The genes are made of DNA. DNA is a very large molecule, and it's made up of these repeating units called nucleotides. There are 4 different bases in the nucleotides: adenine, guanine, cytosine, and thymine. The order in which they're strung together determines the protein that's produced. The shape of DNA is called a double helix, and the bases pair together very specifically. Adenine pairs with thymine, and guanine pairs with cytosine. Like I said before, the order of the bases is what distinguishes different DNA strands and ultimately produces different proteins that do different jobs in our bodies. The proteins are made by combining amino acids. Proteins are big long chains of amino acids. And again, the sequence of those amino acids determines the shape and the function of the protein. Each group of 3 nucleotides codes for a particular amino acid. So GAG codes for the amino acid glutamine, CGT codes for an amino acid called alanine. If there's a mutation, if a nucleotide is changed, then the wrong amino acid can be placed in the protein, and that can completely destroy the protein. This is where we see genetic diseases show up. This is the basis for many diseases and health issues if we have these substitutions or mutations. Before a cell divides, DNA copies itself so that each daughter cell has a copy of the DNA, that code to produce proteins. Replication happens first with the unwinding of the DNA strands of the double helix. Each strand can be exposed to free nucleotides that will be put into the correct position using base pairing. So that's what you see here. The DNA is unwinding, and then these open strands are the ones that have the free nucleotides that come in and base pair with them. There are enzymes and proteins like DNA polymerase that help out in unwinding the DNA, keeping them apart, and assembling the new DNA strands. PCR, which we'll talk about in a little while, is a man-made technique, man-made DNA replication, replicating small quantities of DNA or broken pieces of DNA found at a crime scene. Like I said, man-made in the laboratory. The ability to copy small bits of DNA means that we can take a really small sample of DNA from a crime scene and analyze it. Recombinant DNA relies on these restriction enzymes to cut DNA into fragments. Restriction enzymes come from bacteria, and they're basically highly specialized scissors. They cut DNA molecules at a specific sequence of bases. Once the DNA has been cut, the next step is to put, if we're doing recombinant DNA, it's to put that DNA segment into usually a bacterial cell. And then whatever that DNA that we inserted into the bacterial cell, sometimes it's, for instance, human insulin, We have bacterial cells make human insulin. That's the synthetic human insulin that's made for diabetic patients. Here's a diagram of recombinant DNA. So over here we have bacterial DNA, and then here's the, let's say it's the human insulin gene. That's inserted into the circular DNA of the bacterial cell. That DNA is put back into the host cells, and then it'll divide and multiply and produce human insulin. When we're looking at DNA fingerprinting, which is also known as DNA typing, we look at areas of repeating sequences of bases called tandem repeats. This one is showing a 15 base core, so a sequence of 15 bases that's repeated over and over and over again. These tandem repeats just look like junk, but in forensic science, we can use those to distinguish one individual from another. Tandem repeats are thought to act as fillers or spacers between the coding regions of DNA. They also cap the ends of the chromosomes. All humans have the same type of repeats, but there's variation in the number of repeats each of us has. So this is one person that has 3 repeats, here, here, and here. This is a simplified version of what's really happening. This person over here only has 2 repeats, here and here. So that would distinguish one person's DNA from another. These length differences are associated with long repeating strands. They're called RFLPs, restriction fragment length polymorphisms. And RFLPs were used first in DNA typing. Just like the diagram before showed, a core sequence is 15 to 35 bases long, and it can repeat itself up to 1,000 times. So these are huge chunks of DNA. There are all kinds of different possibilities that exist for the number of repeats a person can have. And that is, again, what distinguishes one person from another. It individualizes people. Once we've broken DNA into different fragments, we use a process called electrophoresis. In this case, it's DNA that's the material that's forced to move across a gel-coated plate, using an electric current. DNA is separated and characterized in this way. Take a look at this diagram, and we're actually going to do this in class also. We have different DNA fragments of different sizes placed into a gel, and then we run an electric current through it. DNA is a charged molecule, so it's repelled by the negative side of the gel, and it moves down the gel, away from the negative end. Smaller fragments move more quickly and they move farther. Longer fragments move more slowly. If you zoomed in on a gel at the microscopic level, we took a cross section of the gel like this. If we zoomed in on it right here, it's like spider webs. It's like a big network of connections. The smaller fragments can move through this web more easily than the larger fragments. So the first step in DNA fingerprinting or DNA typing is the DNA molecules are cut up by restriction enzyme, and then those fragments are loaded into a gel and separated by electrophoresis. Again, the smaller fragments will move faster and farther than the larger ones. And then the fragments are transferred onto a nylon membrane. They're literally blotted onto a nylon membrane, and this process is called southern blotting. So we see southern blotting here, where we left off where the DNA is loaded into a gel, electrophoresis. Now we've blotted that up onto a nylon membrane. We treat the nylon membrane with radioactive probes. This is called hybridization. DNA is not able to be seen by the eye, so we have to make it radioactive and then take an X-ray of it. Now we can see the banding patterns. So like I said before, we have X-rays. We literally take X-rays of the radioactive probes will make the bands appear. A typical DNA fragment will show two bands, one restriction fragment length polymorphism, one RFLP, from each chromosome. When you compare the DNA fragment patterns from two or more people, you look for a match between the band sets. You can use a lot of different probes and combine their frequencies to figure out very specifically who the person is. So two or more specimens can come from two different people or from the same person. If they come from the same person, they're a match. So back to PCR, this man-made DNA replication, which allows us to take a really small DNA sample and make it bigger so we have enough of that DNA to analyze it. PCR is used to amplify really small pieces or really small amounts of DNA many millions of times. So this happens in sort of 3 easy steps. First, the DNA is heated. Anything that is heated expands, so DNA is heated to separate the two sides of the DNA strand. And then primers are put in, like here, that hybridize or base pair with the existing strands of DNA. Then we add in DNA polymerase, which makes new DNA, and the free nucleotides, just like the process happens in a cell. These are used to rebuild each of the separated strands. And this process is repeated over and over again, 25 to 30 times. Since we're doubling the DNA every time, it's exponential growth. It's a really fast process. PCR can't be used with these big old chunks of DNA, the RFLPs. RFLP strands are too long. Like we said, thousands of bases. Their repeating sequences are 15 to 35 bases long. PCR is best used with DNA that's no longer than a couple of hundred bases. So we needed a new type of DNA repeating sequences to use. So if we're using shorter DNA strands, they wouldn't break as easily. They'd be more stable. They don't break down as easily. So they don't break apart. They don't break down in the environment if there's bad environmental conditions. Long RFLPs break apart under adverse conditions, which are not uncommon at crime scenes, like bad weather or rain or what have you. PCR also has an advantage, again, that it can amplify really small quantities of DNA. So the new repeating sequences that are used are called short tandem repeats, STR analysis. These are short sequences that repeat themselves within the DNA molecule over and over and over again. They're useful for identification because they're found in great abundance throughout the human genome. These are only 3 to 7 bases in length, and the entire strand is less than 450 bases in length. So because they're smaller, they're more compact, they don't break down as easily, and they can be recovered from bodies that are extremely highly decomposed. They're also easier to amplify by PCR. There are hundreds of different types of STRs that are found in human genes. The more STRs we can use, the smaller percentage of the population from a combination of STRs can come. So we really get to individualize a person and create a unique DNA fingerprint. This is called multiplexing. So we can take out and amplify a combination of different STRs. And you see here where these banding patterns match. These two DNA samples came from the same individual. U.S. crime labs have 13 STRs that are used to identify people. The database that's used to house all these DNA fingerprints is called CODIS. It's the National DNA Database. Individualization can come from analyzing these combinations of STRs and determining the product of their frequencies. With an STR, as little as 125 picograms of DNA is required for analysis. This is 100 times less than is required for RFLP analysis. CODIS it stands for the Combined DNA Index System. It is, like I said, where all DNA fingerprints are housed. This is from crime scenes, convicted offenders, and profiles of missing persons. Currently, not everyone is required to have their DNA in CODIS. Finally, how do we package biological evidence? We've talked about this before. Just to review, we need to photograph and sketch that evidence. We need to wear gloves. Clothing that has blood evidence has to be collected. And then it's packaged, remember, in paper bags. Packaged separately in paper bags. Because if we package in plastic, bacteria and fungi can degrade the sample. Dried blood is removed using a moistened sterile cotton swab with distilled water, and then put in a swab box or paper or manila envelope. Everything should be refrigerated to prevent spoiling, and then standard reference samples have to be collected. Blood samples or buccal swab, which is swabbing the mouth and the cheek, the inside of the cheek.